Okay, we are back with the eukaryotes or the eukarya, and these are going to be our organisms that have a nucleus, right? So a nice membrane-bound nuclear envelope containing their genetic material. So what I put up here, right? So the eukarya, you can see on the side of the phylogenetic tree here, one of the domains of life. If you kind of zoomed in on that and looked at the diversity of eukaryotes, that's what this tree down here is. Now, you are probably familiar with plant, animal, fungus, protist, being the kingdoms within this domain, but that's changing, and you'll actually notice that in lab. Your lab manual is now arranged based on these new, what they're calling supergroups. Basically what scientists realize, right? The whole idea of grouping organisms is to make sure any group you create, whether that's a genus or a family or a kingdom, that the organisms inside of that group are more closely related to each other than they are to any other organism. Well, that didn't happen, right? So as we've started using DNA analysis a lot more, people started noticing, man, there's some organisms in this group, maybe protist, that are actually more closely related to plants. And so protist is a terrible group. I will tell you that. Protists are basically eukaryotes that are neither plant, animal, or fungus. It was kind of the grab bag. And so we're starting to kind of spread those organisms out. So I don't know how quickly this is going to happen, but kind of tuck that in the back of your mind, that some of this terminology might be changing, right? Basically, you'll find Apistacantha. That's where you're going to find all the animals and other organisms that are closely related. So that'll be fun, right? Okay, so just a reminder here in microbiology, we're looking at eukaryotes, but they're going to be small eukaryotes. Some of them are still multicellular, even though they're small. So we have a little bit of variety depending on what group we're looking at. You probably talked about this in Gen Bio, but just a quick reminder, in this chapter, they talk about the theory of endosymbiosis. And this is the idea that an organism, and they did a nice job here, they're calling it a pre-eukaryote. Notice it's not a bacteria engulfing another bacteria, but it's this like ancestor of a eukaryote that did engulf a bacteria. They had a symbiotic relationship, maybe one where both individuals benefited, and so sure enough, that relationship sticks around. So the organelles that probably have the strongest evidence for this are going to be the mitochondria and the chloroplasts, both of our energetic organelles. They have their own DNA. That DNA looks just like bacterial DNA, as in it's a single circular chromosome, that sort of thing. So there is some really good evidence that this comes from kind of an ancient relationship that took place, right? And then we see here in our eukaryotic cell that that nucleus is now membrane bound, and sure enough, those bacteria are now considered organelles. It's pretty fascinating if you look into that a little bit more. Okay, so I pulled up this chart. It looks just like the one we used on the bacteria in the previous chapter, but this is going to be super quick. This is totally review from Gen Bio. So just a couple things I want to point out. If we look at the appendages, right, we only have two now. We have flagella and cilia. These are both going to be for locomotion. Now, the flagellum in eukaryotic cells has the same role, right, to move the organism around, and superficially they look very similar. But if you get down into kind of the microscopic anatomy, they're actually set up fairly differently. And what you'll see here in the eukaryotic cell is that this flagellum beats, where the prokaryotic bacterial flagellum kind of rotates, if you remember those runs and tumbles. And so this would be an example of what we call convergent evolution, where the structure looks really similar, but it's probably not due to common ancestry. This is a solution that maybe arose multiple times over the history of life. Cilia, they're basically short flagellum. There's lots of them on the surface of the cell. They're all beating. If you're talking about a single-celled organism, like we often would be here in micro, this is for movement. In our trachea, the cell is stuck right on the side of the trachea, and the cilia are beating, and it's moving mucus past the cell. Now we're actually looking at this as an opportunity for our eukaryotes to move around. But notice we did not talk about cilia in the bacteria. No bacteria have cilia. This is only found amongst eukaryotes. The glycocalyx, that sugary protein, kind of gelatinous, it could be a slime layer, it could be a capsule. We tend to skip that in Bio 111. We kind of tend to skim over that, but it does exist in eukaryotic cells as well. It's actually really important, probably in helping protect cells from dehydration, particularly if you're a unicellular organism. I am not going to waste your time by going over the cell membrane again, right? You know it's a phospholipid bilayer studded with proteins that's a selectively permeable membrane. I will point out the cell wall, because this varies dramatically depending on which kingdom or supergroup you're in. No animals have a cell wall around their cells, right? So humans do not have cell walls. Plants obviously have cell walls. Those are made out of cellulose. Fungi have cell walls made out of chitin. And then when you get into the protists, particularly when you get into the protozoans, particularly the algae, you get a lot of diversity. There's some that use basically glass to make cell walls, cellulose, pectin, all sorts of different things. So there's a big variety there. Now, as far as inside of the cell, you should recognize a lot of these organelles. And so what I'd like us to do in class is we're going to take some time kind of reviewing these, but I don't want to stand here and talk at you. But if you are not remembering that, or you didn't go over those in Gen Bio, hop on Khan Academy, hop on Crash Course Biology, do a quick review. Your book also reviews them, and that way you can come to class and be prepared to contribute to our review. All right, see you there.